Hello, we are Senior Lab Group 10, and our video is on the Bioreactor Lab. The purpose of this lab is to compare the kinetic parameters for bacterial growth of the LB media and M9 media. We wanted to measure the bacterial optical density, OD, over time to construct a bacterial growth curve and estimate the substrate concentration in our reactor as time progresses. We then compared our data with previously collected data for both the M9 growth media and the nutrient-rich LB media.
Here are some important equations that we need for this lab. X is the biomass concentration, S is the substrate concentration, YXS is the maintenance coefficient, which was 0.5 for this experiment, mu is the growth parameter, Ks is the monad constant, and mu max is the maximum growth rate parameter. Ks and mu max will be calculated experimentally, which can then be used to calculate mu. X and S will be solved with respect to time. To model the growth of E. coli with respect to time, OD measurements will be taken every 30 minutes for 6 to 8 hours. This is done with a UV-vis spectrophotometer with the wavelength set to 600 nanometers.
The main idea of this experiment was to set up a laboratory-scale bioreactor and determine the biochemical kinetic parameters such as biomass, substrate concentration, and the monad constant. On the first day of the bioreactor lab, our group first prepared 3 liters of liquid broth media according to the instructions on the bottle. We carefully weighed out 20 grams of the solid LB media and combined it with 1 liter of deionized water to obtain the desired concentration of 20 grams per liter. We repeated this process 3 times to obtain a total of 3 liters of LB media. Before we began the experiment, we ensured that all the glassware was properly sterilized by the autoclave steam to avoid contamination. Our group collected the necessary equipment into the basket that would be inserted into the autoclave, ensuring to wrap with aluminum foil at the openings of all the equipment and taping with special autoclave tape. The autoclave was set at 120 degrees Celsius for 15 minutes. This autoclaving process was repeated for the bioreactor. On day 2, we prepared the bioreactor. This procedure required us to set up the afternoon prior to the actual experiment. The reason for this was to polarize the dissolved oxygen probe as well as inoculate the bacteria for the shaker flasks. We rinsed the dissolved oxygen probe with DI water and again with ethanol to clean it. We then carefully placed the probe into the ring stand inside the biosafety hood in order to sterilize it with UV light for 5 minutes. The dissolved oxygen probe was then placed into the bioreactor overnight to polarize. To inoculate our solutions, we first added 2 liters of our media to the bioreactor.
We then added 60 milliliters of our media into 2 150 milliliter flasks. After that, we added 0.5 milliliters of E. coli bacteria into each of these flasks and set both of them on a shaking table at 37 degrees Celsius and 200 RPM overnight. We only utilized one of the flasks when we measured data, but the second flask was just a backup in case the bacteria cells did not incubate well overnight. Growth is indicated by cloudiness as a result of respiration from the bacteria.
To start day 3, we calibrated the pH probe by rinsing it with DI water and placing it into a neutral buffer solution for 5 minutes, calibrating it with the bioreactor. We then rinsed it off and placed it into an acidic buffer solution for 5 minutes and again calibrated it with the bioreactor. We started our experiment by adding 2 liters of our LB media into the bioreactor. We then calibrated the DO probe using the bioreactor control system. To start the bioreactor, we primed the temperature control setting before setting it to PID. While the control system was rising to 37 degrees Celsius, we blanked the spectrophotometer at 600 nanometers to prepare to take measurements of our samples. This is important as the optical density at 600 nanometers will determine our cell concentration. To begin our experiment, we added 50 milliliters of our inoculated bacteria into the bioreactor at our initial time point of 10 a.m. Every 30 minutes from then on, we collected 1 milliliter of our sample using a sterile syringe and measured the absorbance, pH, DO, and time. We then started our experiment for the benchtop shakers, filling each of the 250 milliliters flasks with 100 milliliters of LB media. We would then pipette 2.5 milliliters of our inoculated bacteria into each of the flasks. This was our initial time point at 10:15 a.m. We micropipetted 1 milliliter of our sample, ensuring to change the tip for each measurement and use a different cuvette for each flask to measure absorbance. We continued taking measurements at every 30 minutes until 2:45 p.m., ensuring to record the absorbance and time of each measurement and empty the waste into the bio-waste container after, and also making sure to put the flasks back onto the benchtop shaker after each iteration.
When cleaning up the experiment, we made sure to rinse everything with antibacterial soap and water. We poured bleach into the bioreactor and waste container that had bacteria before pouring it down the drain. We wiped the countertops with 70% ethanol solution and disposed of our gloves into a separate waste container.
We then determined mu-max for the LB media used in our experiment. This involved plotting our bioreactor data and focusing on the initial 3 points, which correspond to log phase, the period of fastest bacterial growth. Through linear regression analysis of these points, we established a slope equivalent to mu-max, which we calculated to be 0.0234 per minute.
We derived the Monod constant for the M9 media by leveraging data provided by another group. This process began with plotting the given M9 data against time, followed by conducting a polynomial regression and subsequently calculating its derivative. Denoting biomass concentration as x and time as t, we obtained dx dt for each data point. Utilizing equation 2, we computed mu for each point, then proceeded to determine s at every instance using equation 3. Having found mu and s values, we also derived 1 over mu and 1 over s for each point. Subsequently, we plotted 1 over mu against 1 over s and performed linear regression, yielding an equation similar to equation 4. Within this equation, the slope contains the Monod constant that we were looking for, which we can calculate after we calculate mu max.
Overall, we conducted our experiment in order to compare the results of the M9 data to the LB bioreactor data to the LB shaker flask data. We found that the growth rate for the M9 media was slower than the growth rate for the LB media based on the graph showing cell concentration versus time. The mu max for the LB bioreactor had a value of 0.0234, whereas for the M9 data, that value was around 0.0035, showing that the LB bioreactor had approximately 10 times higher growth rate. We also compared the absorbance values between the bioreactor LB media and the shaker flask. For example, the bioreactor values were lower by roughly 1/2 in comparison to the shaker flasks. The temperature gradually remained at 37 degrees Celsius for both the flasks and the bioreactor. However, the flasks were removed from the benchtop shakers when taking samples, whereas the bioreactor continued mixing.
This implies that the flasks did not experience consistent mixing throughout their time on the heated shaker bed. One thing to note is that the dissolved oxygen reading stayed at 100% for the entirety of the experiment, which is most likely incorrect. However, the bioreactor control system is quite old and may no longer be able to produce accurate readings. In conclusion, this bioreactor experiment demonstrated that the kinetics of a bioreactor cell growth are highly dependent on several variables such as time, temperature, mixing speed, and media type. We would like to thank our TA, John Grasso, and Professor Lay for helping us with this experiment and throughout this semester.